Hi everyone and welcome back again to Biology Circuit. In this video I am going to discuss some important questions from genetics unit of CSI NET. So let's start. The first question: recessive lethal alleles are never completely eliminated from the population because option 1 lethal alleles are always conditional in nature, option 2 lethal alleles have selective advantage, option 3 lethal alleles protect organisms from other deleterious mutations. Option 4, they are maintained in the population as heterozygotes. So the right answer will be option 4. They are maintained in the populations as heterozygotes. Next, a positive association between absolute average individual fitness and population size over some finite interval is known as option 1 allele effect, option 2 founder effect, option 3 Lenz's rule, and option 4 Bergman's rule. Right answer will be option 1 allele effect. Next, the following is the inheritance pattern of a trait under observations. The trait often skips generations. The number of affected males and females is almost equal. The trait is often found in pedigree with consanguineous marriage. The trait is often found in the pedigrees with consanguineous marriage. The trait is like to be autosomal recessive, autosomal dominant, sex-linked recessive, sex-linked dominant. The right answer is option 1, autosomal recessive. See, the first characters is told the trait often skips generations. So while it is skipping generation, it cannot be the dominant one. So it is recessive. The number of affected males and females almost equal. So it is not sex-linked. It is not biased. And the third is the trait is often found in pedigrees with a consanguineous marriage. So the trait is likely to be autosomal recessive. It's a very good question and important also. Next, the C-strands complementation test is carried out to identify option 1 if two mutations are allelic in nature, option 2 if two genes interact with one another, option 3 the number of genes influencing a phenotype, option 4 to understand the dominance and recessive relationship between alleles. A C-strands complementation test is carried out to identify. The right answer will be option 2 if two genes interact with one another. Then C-strands complementation test is carried out to identify. Next, the degree of genetic relatedness between the offspring and their parents is option 1 higher than that between sister and brother, option 2 lower than that between sister and brother, option 3 the same as that between sister and brother, option 4 dependent on the number of siblings. So the question is the degree of genetic relatedness between the offspring and their parents is right answer will be option 3, the same as between the sister and brother, the degree of genetic relatedness. So between the parents and offspring and between the sister and brothers, the degree of relatedness, genetic relatedness, it is similar. Okay, move to the next. The dynamics of any subpopulation within a metapopulation differs from that of a normal population in that the option 1 birth rates are lower than the death rates, option 2 death rates are lower than the birth rates, option 3 immigration and emigration rates are significantly higher, option 4 immigration and emigration rates are negligible. So the right answer will be the immigration and emigration rates are significantly higher. The dynamics of any subpopulation within a metapopulation differ from that of a normal population in that the immigration and emigration rates are significantly higher. Move to the next. Directional selection for a particular trait will lead to the frequency. Option 1 being normally distributed in the population, option 2 always showing a left skewed distribution in the population, option 3 always showing a right skewed distribution in the population, option 4 showing either a right or a left skewed distribution in the population. Right answer is the option 4. Directional selection for a particular trait will lead to the frequency showing either a right or a left skewed distribution in the population. Moving to the next. Which one of the following statements regarding molecular markers for genotyping is incorrect? Option 1. Polymorphism in intronic regions of a gene cannot be used for trait mapping. Option 2. Codominant molecular markers can be used to detect heterozygosity. Option 3. Sequence stacked microsatellite sites and simple sequence repeat polymorphisms are based on polymorphism in repetitive DNA sequences. Option 4. Restriction fragment length polymorphisms, that is RFLPs, and simple sequence repeats SSRs are multi-allelic markers. So the right answer will be option 1. That is the incorrect statements regarding molecular marker for genotyping. Polymorphism in intronic regions of a gene cannot be used for a trait mapping. Okay, because it is an intronic regions. Next. Which one of the following conditions associated with chromosome 15 may cause Prader-Willi syndrome? Option 1. Paternal uniparental disomy. Option 2. Maternal uniparental disomy. Imprinting of 15q11, 15q11, q13 locus in maternal copy. Imprinting of 15q23, q25 locus in parental copy. So the right answer will be maternal uniparental disomy. That is associated with the chromosome 15 may cause Prader-Willi syndrome. Okay, next. Reduction in the frequency of heterozygous genotype with a concomitant increase in the frequency of homozygous genotype in the context of random mating is due to genetic effect, genetic drift, intense inbreeding, reverse mutations, founder effect. So the right answer will be intense inbreeding. Answer 2. Next. A colorblind father has a daughter who is also colorblind and has Turner syndrome. The genotype of the daughter is due to translocation event in father, translocation event in mother, non-disjunction event in mother, non-disjunction event in father. So the right answer will be option 3, non-disjunction event in mother. So if you like this video, please subscribe to Biology Circuit and share it. That's all for today. And keep watching Biology Circuit for more such videos. Thank you.